Hello and welcome back to Suchitra's Biology. Hope you all are doing good. So today we are going to discuss about the topic that is the GPCR NO signaling. So let's start. GPCR NO signaling, basically it helps in vasodilation. Okay. Vasodilation is what? Vasodilation is increase in the diameter of blood vessels. Vasodilation is what? That this is the increase in the diameter of the blood vessels which causes increased blood flow. Okay, increased blood flow. Okay, so we know the blood vessel is made up of 3 layers. Okay, this is the outer layer called tunica externa. This is the middle layer called tunica media, middle layer, and this is the innermost layer known as tunica interna. Okay, now tunica media is made up of smooth muscles, and tunica interna is made up of endothelial cells. Tunica interna is made up of endothelial cells. Okay, so this GPCR NO signaling basically requires this tunica media and the tunica interna. Okay, these 2 layers. So let's discuss how this GPCR NO signaling occurs. Suppose this is this one is endothelial cell. Okay, endothelial cell, that is the interna, tunica interna layer. Okay, and this is smooth muscle layer, that is tunica smooth muscle layer. Okay, that is tunica media. So GPCR NO signaling occurs in between these 2 layers of the blood vessel. There is GPCR present, same GPCR 7. This is the GPCR. This is the G protein, alpha, beta and gamma. Okay, it is bound to GDP. G protein, this is the GPCR, and this is the G protein. G protein consists of alpha, beta and gamma subunits, and alpha subunit is bound to GDP when G protein is in inactive form. Okay, when GTP binds to the alpha subunit of G protein, it is said to be in active form. When a ligand comes and binds to the receptor, this ligand is acetylcholine. Okay, so I was telling that when the ligand comes and binds to the GPCR becomes active, and what the GPCR does, it removes the GDP from the alpha subunit and adds GTP to it. Okay, this is GTP. Once GTP is bound to the G protein becomes active. Okay, this alpha GTP will now dissociate from this gamma and beta subunit of G protein. This alpha subunit will dissociate from the gamma and beta subunit of G protein, and it will bind to PLC. Okay, it will come and bind to PLC, that is phospholipase C. Phospholipase C. Okay. Now once it comes and binds to the PLC get activated. And now what PLC will do? PLC will convert PIP2 into DAG and IP3. PLC does what? PLC gets activated when the alpha GTP is bound to this. Now PLC converts PIP2 into DAG and IP3. Okay. DAG and IP3. Now this IP3, this is the ER. Okay. Endoplasmic reticulum or sarcoplasmic reticulum. It has calcium ions in it. And on the membrane of ER or sarcoplasmic reticulum, there is a channel called IP3 calcium gated channel. When IP3 will come and bound to this or bind to this, the channel opens and it will cause release of calcium 2 plus in the cytosol. Okay. IP3 comes and binds to the channel and causes the opening of the calcium channel present on the ER, and calcium comes out. Now calcium binds with calmodulin. Okay. Calcium combines with calmodulin to form CC complex. C for calcium and C for calmodulin. So it forms CC complex. Now this CC complex combines with CAM kinase and it forms triple C complex. Calcium, calmodulin, CAM kinase complex. This CC complex is what? This is kinase basically, no? CAM kinase. So this is kinase and it can phosphorylate any protein. So it will convert NO synthase to active NO synthase. This was inactive and this is active. This CCC complex phosphorylates or adds phosphate group to this NO synthase, and now NO synthase becomes active. Now NO synthase will convert arginine amino acid into citrulline plus NO. Okay, citrulline and NO. GPCR was there. Acetylcholine, a ligand, comes and binds to the GPCR. It causes the GTP binding to the alpha subunit. Now alpha subunit with GTP goes and binds to the PLC, phospholipase C. Now activated PLC converts PIP2 into DAG and IP3. This IP3 comes and binds to the ER membrane, binds to the channel on the ER membrane. Okay, that causes the opening of the channel and the release of the calcium 2 plus ions into the cytosol. Now calmodulin combines with Ca2 plus to form CC complex, calcium calmodulin complex. Now this CC complex combines with CAM kinase to form triple C complex, that is calcium calmodulin CAM kinase complex. Now this activated CCC complex, it will phosphorylate the NO synthase to phosphorylated form. Okay, that becomes active. NO synthase become active on phosphorylation by CCC complex. This active NO synthase will convert arginine amino acid into citrulline and NO. Now this NO is a gas basically, right? And gases can diffuse through the plasma membrane. Okay, so this diffuses to the smooth muscle or the tunica media layer. Okay, the NO is basically synthesized in the endothelial cell or the tunica interna layer, and this NO then diffuses into the tunica media or smooth muscle layer. Now what happens? Let's see. This NO is present here. Okay, it diffused and it goes here. Now this NO will activate guanyl cyclase. Guanyl cyclase. Okay. This is guanyl cyclase. NO goes into the smooth muscle layer and it binds to the guanyl cyclase and activates guanyl cyclase. Now guanyl cyclase becomes active. So active guanyl cyclase converts GTP into CGMP. Okay. GTP, guanosine triphosphate, into cyclic guanosine monophosphate. Okay. CGMP is nothing but the two phosphates are removed and it forms a circularized structure. So it is cyclic guanosine monophosphate. Now the CGMP activates PKG, that is phosphokinase G. Okay, this is phosphokinase G. As PKG gets activated by CGMP, now PKG will cause vasodilation. This was the main aim of the GPCR NO signaling. Vasodilation will occur means the diameter of the blood vessels. If this is the diameter of the blood vessel, okay. Now due to vasodilation, the diameter of the blood vessel will increase. Now if the diameter of the blood vessel increase, more blood will flow now. So blood circulation will increase due to vasodilation. Okay. So this was the GPCR PKA signaling. Now let's see the application of the GPCR NO signaling. Okay. A medicine known as commonly as viagra, okay, is used a medicine called viagra is used. It is used by patients that have problem in penile erection. Okay, so viagra increases blood flow. It increases blood flow, so the patients who have problem in penile erection, their problem is cured. Okay. Now let's see how it causes vasodilation. We have seen that CGMP is the main thing that is required to activate the PKG, okay, phosphokinase G. And once PKG is activated by CGMP, PKG will cause vasodilation and increase blood flow, right? Now the CGMP gets converted to GMP, okay, by enzyme called phosphodiesterase. Enzyme called phosphodiesterase CGMP. It converts CGMP into GMP. As CGMP is converted to GMP, CGMP levels are low. So low CGMP levels will not activate PKG, and if the PKG is not activated, vasodilation will not occur. Okay. So this Viagra medicine blocks this. Viagra, what it does? It blocks the conversion of CGMP to GMP. Okay. It does not allow the CGMP to convert into GMP. And if it does not allow the CGMP to get converted into GMP, now the CGMP levels are high. Now as CGMP levels will be high, it will cause activation of PKG, phosphokinase G, and if the phosphokinase G are activated, vasodilation will occur. Vasodilation, dilation. Okay. Vasodilation means increased blood flow. And so Viagra helps in continuous penile erection. Okay. It helps to solve the problem in patients that have a problem in penile erection. Okay. So this was all about the GPCR NO signaling. If you like the video, do like, share and subscribe my channel. Thank you.